Okay, while we're waiting for students to join, are there any questions? Let me start by showing you our schedule. We'll be working on Chapter 4, Prokaryotic and Eukaryotic Cells today. And we might get off early. I've decided I don't want to go ahead with Chapter 5. And today it's a long lecture day, and so we'll finish Chapter 4. Tomorrow we'll start Chapter 5 and Lab 3. All right, any questions about the schedule? Today, if you want to take the practice quiz, you must take it by 11:59 p.m. tonight. It would be a good idea to get a practice in on before you take the real quiz on Friday. I'll give you an extra credit point as well. I had something else to say. If we look at the schedule, can you guys see this? There it is, we're at week 2, Chapter 4 notes, and at the end of this, there's extra study. Week 2, what you need to do is get together with some other students. You can do it online. You can do it over the phone. I do not expect you guys to get together. And just follow the directions. Turn in a list of the people who you studied and give me the full names. And the study session has to be for half an hour. A few other requirements to turn in. Just tell me what you're doing and when you study, submit it, and then I'll give you an extra credit point per week. There will be a link like this for each week. I sent out an email about that, and you just put it up in the folder in the Canvas. I don't think there's anything else I want to talk about. Are there any questions?
What is the Respondus lockdown browser?
Yes, I haven't quite decided about that. I'll look into it. I'm checking with the biology department to see if they approve of this and what their experience is with it. But this means that when you take quiz 1B, if I am requiring, and I haven't made the decision yet, and I don't have to make it until June 11th, really, but I'll make it beforehand. It means that when you take the quiz, when you open up the browser and you start the quiz, it will lock down the browser so that no other windows can be open. And you will be locked down in the browser until you finish the quiz. The purpose of that is just to reduce on cheating and
So you don't have to download anything extra?
No, you don't have to download anything extra.
Okay, thank you.
It is a downloaded. It is a download. I have it for my anatomy class, which people are still having problems trying to get it to work. So my professor nixed it for the first quiz, but it is a download for your computer.
I was told that it's not a download, it is just something that runs with the quiz. I could have you taking the camera where it'll take your picture, and now that probably would be a download, but all of you must have a camera because you're on Zoom. But I decided not to have that, so mine would only be the browser lockdown. Anyways, like I said, that isn't decided. I will get back to you about that. But I'm thinking of trying it all it'll do, like I said, is lock down your browser while you're taking the quiz. The bad part is that if for some reason you want to leave the quiz, I don't think it will let you leave the quiz until you hit the submit button. Okay, and that's not really a big problem because I don't think most of you will be wanting to leave the quiz before you're finished. But like I said, this isn't totally decided, but I might be requiring a respondus lockdown of the browser. Okay? I guess I should have done it on the practice quiz and then we can see this. If somebody wants, I can make another practice quiz and you guys can go through the respondus browser lockdown on a practice quiz. But anyways, let me get back to you on that because the decision hasn't been totally made. And when I hear back from the biology department, I will let you know. All right?
Thank you.
Do remember that quiz 1B, don't ask me why I called it quiz 1B. Quiz 1 will be due by 11:59 p.m. June 12th, that's Friday. You must take it between 11:59 p.m. Thursday and 11:59 p.m. Friday. And it's worth 50 points. Okay. Any other questions? If not, let's go ahead and get started with the lesson. I believe the last slide I talked about was the plasma membrane last time.
That's correct.
Okay, thank you for correcting me. So let's continue talking about the plasma membrane. I can probably blow that up a little. The plasma membrane, we have a model of it and it's called the fluid mosaic model. It's a model on how the plasma membrane works. And what it states is that the membrane is a mosaic of phospholipids and proteins. So they're together sort of like this. Let me blow that up a little better. In the cell membrane or the plasma membrane. And when we say it's a fluid mosaic model, the membrane is as fluid as olive oil. And if you worked with olive oil, you know it's pretty fluid, at least at room temperature or 37. And it's kind of amazing because when you see my hand here, it's being held together only by cell membranes and of course the skin. And that is a fluid, which to me, that's pretty amazing because that feels pretty solid. The proteins, generally speaking, move about in the fluid membrane. If you take an anatomy and physiology and you learned about receptors, the receptors, once they bind with a ligand, what stimulates the receptors move together. So that tells you that the proteins can move in the cell membrane. Most of the proteins do move, not all of them do. The ones that do not are the ones that are anchored in place by something, such as if this protein were anchored to the cellular cytoskeleton inside the cell, then this protein, then this protein would not move in the fluid membrane because it's anchored in place. But this protein and that protein, and probably that protein and that protein, they're not anchored by anything. And so they're moving about in the membrane. And that also applies to the phospholipids. They will rotate in place, spinning around, and they will also move laterally. Okay, so moving around. Any questions about the fluid mosaic model of the cell membrane? All right. So the plasma membrane is selectively permeable, meaning it allows certain things to cross and prevents other things from crossing. Now that creates a problem for the cell because the cell generally wants molecules coming into it and then waste molecules to leave it. Like CO2 is generated by the cell, and that's a waste molecule and it has to leave. But the cell membrane does allow certain molecules to cross. It allows the passage of some molecules, and that's why we call it selectively permeable, but not others. No large molecules can cross the cell membrane without a transport protein. Now, sugars like glucose and amino acids, the cell is going to want those to come into the cell. And what the cell does is it makes a transport protein to bring those molecules across the cell membrane. And generally speaking, for each different molecule, like glucose or the amino acid methionine, the cell will make a specific transport protein that brings that one molecule across the cell membrane. Is that clear? So generally speaking, transport proteins are specific for one molecule. There are some exceptions, but generally they are specific for one molecule. Ions either do not cross the cell membrane on their own, or they cross very slowly. So once again, if the cell wants this to come into the cell, like the chloride ion or the potassium ion, the cell has to make a transport protein to bring that molecule across. Lipid-soluble molecules can cross the cell membrane. But if you think about a red blood cell in the blood, there aren't that many lipid molecules in the blood because the blood is mostly water. And so lipids do not generally come into the contact with the cell membrane and then readily cross. They can do that, like if you eat a lipid-rich meal, the lipids will be put into the blood and then it will go into the cells. But I'm just saying that, well, let's just end it at lipids can cross the cell membrane because the cell membrane, what's being, boy, that really moved. I tried to move it one slide and move many. What's acting as the selective permeability is the fatty acid tails of the lipid bilayer. And lipids can get into that and then just cross the cell membrane. Smaller molecules can also cross the cell membrane, especially if they're nonpolar, like oxygen and carbon dioxide readily diffuse right through the cell membrane. And that's important for cells because oxygen has to come in and carbon dioxide has to leave. And so they move into and out of the cell simply by diffusion. Now, water is a small molecule, but it is polar. It can cross the cell membrane, but not very easily. And what happens is that what water does is, if there's more water outside the cell than inside the cell, water wants to diffuse into the cell, and it can come up to the cell membrane and then wait for a phospholipid to move over, and then water can sneak in between the phospholipids. But it can't get through the next phospholipid, and so it just waits there. And then eventually a phospholipid moves over, and then water can move up and then water can move into the cell. And so water can move into the cell through the plasma membrane, but it doesn't do so very readily. And the cell, like plant cells in the soil, generally want water to come into the cell very readily. And so they create transport proteins to bring water into the cell. Any questions about the plasma membrane and selective permeability? The plasma membrane also contains a number of enzymes in it, and the one I want to talk about, we'll talk about it in a future lesson, not today, is that in bacteria, as well as archaea, but we're not going to talk about archaea. So in bacteria, the plasma membrane has the enzymes for the bacteria to make ATP. That does not happen in the plasma membrane of eukaryotes, because if you do recall from whatever your previous class was, I'm sorry, I don't know. I just got hired by Yavapai and I don't remember what was the class that was a prerequisite for microbiology. But when you had that class, and I think there's actually 3 you could take, so one would be Biology 100. Hopefully you learned that eukaryotes make ATP in the mitochondria. So that's why eukaryote cells do not have the enzymes in the plasma membrane for making ATP. And that's because those enzymes are in the inner mitochondrial membrane. For the bacteria, they also have the photosynthetic pigments if they are photosynthetic bacteria. And we call these, which you can sort of see here, chromatophores or thylakoids. And those are infoldings of the plasma membrane where the cell has the photosynthetic pigments for undergoing photosynthesis. Once again, eukaryote cells do not have the enzymes and pigments in the plasma membrane. They would have them in the chloroplast. All right. Any questions about any of that part? Now, if the plasma membrane is damaged, the cell contents, like the cytoplasm, can leak out of the cell, and that would kill the cell. And that damage can happen with alcohols, quaternary ammonium compounds, polymyxin antibiotics like polymyxin B, and they put holes in the plasma membrane. Alcohols, of course, are very hard on lipids, and that's why the plasma membrane gets damaged with alcohol. The same thing with quaternary ammonium compounds. These are found in hospitals. I don't think quaternary ammonium compounds are very common in a house, a normal common household. Polymyxin B is an antibiotic, and what it does is it puts a hole in the cell membrane, and that kills the cell. And we'll talk about all of those later in the term. Oops, sorry. I'm supposed to go down 1. There we go. Movement across the membranes. We're talking mostly about the cell membrane because we're talking about bacteria, but when we're talking about eukaryotes, this can also apply to movement across the membrane of an organelle, like the mitochondria or the endoplasmic reticulum. Cells use 2 processes for having movement across the membranes. There's passive processes, and this is the movement of molecules down a concentration gradient, and we're talking about solutes mainly. and that is solutes move from areas of high concentration to areas of low concentration. And that's passive. It's just the simple diffusion, and it costs the cell no energy when the cell uses the passive movement process. There's also active, and that's when a molecule moves against its concentration gradient, meaning the molecule, if it's moving into the cell, it's moving from areas of low concentration to areas of high concentration. And that costs the cell energy. And to do that movement, the cell uses a transport protein and it expends ATP to move the molecules from areas of low concentration to areas of high concentration, such as when the cell has lots of glucose in it, and outside of the cell, there's a low concentration of glucose. The cell still wants to bring the glucose into the cell, and so it will move it against its concentration gradient. And that's an active process requiring a transport protein, and the cell has to expend energy to do it. Now, in the passive processes, where the cell is moving a molecule from areas of high concentration to an area of low concentration, the cell can do that by three methods. Simple diffusion, and we're just talking about solutes, move from areas of high concentration to areas of low concentration. Already mentioned that oxygen is, generally speaking, higher outside the cell than inside the cell. So oxygen moves through the cell membrane and into the cell from areas of high concentration to areas of low concentration by simple diffusion. Any questions about that? Facilitated diffusion is similar, but different. And once again, molecules move from areas of high concentration to areas of low concentration, but they cannot move across the plasma membrane directly. What they do is they have a channel protein, and the protein allows a molecule to move across the cell membrane into the cell. This is a channel protein, meaning it just creates a channel for the molecule to move by the laws of simple diffusion. It is not a transport protein where it is actively spending energy to move the molecule against its concentration gradient. Any questions about facilitated diffusion? Now, with simple diffusion and facilitated diffusion, we're talking about solutes, molecules other than the liquid, which is the solvent, moving from areas of high concentration to areas of low concentration. When we're talking about a solvent moving in a passive process from an area of high concentration to an area of low concentration, that is not called diffusion, it's called osmosis. And you just have to live with it. There's a specific term for the movement of water across the plasma membrane or any membrane. And we call that osmosis when water moves from areas of high concentration to areas of low concentration across a membrane. But it does follow the laws of diffusion, simple diffusion. But osmosis is always involving a solvent molecule moving across a semi-permeable membrane. So if you had, I don't know, a container of soil and it's very dry over here and you add water to this side, the water would move this way by the laws of simple diffusion. And that's actually a solvent following the laws of simple diffusion. because it's not crossing a semi-permeable membrane from this area to that area, we don't call that osmosis. The water is moving by simple diffusion. Any question about that? Osmosis is when water moves across the semi-permeable membrane by the laws of diffusion. So let's talk a little bit more about simple diffusion. It's the movement of a solute from an area of high concentration to an area of low concentration. Like if I were to have a beaker of water and I added a drop of dye in there, this would be where the dye would have high concentration, and this would be where the dye has low concentration, and the molecules of the dye move out from areas of high concentration to areas of low concentration. And as I stated, in cells, it's important for oxygen and carbon dioxide to move by the laws of simple diffusion. Carbon dioxide moving out of the cell, and then oxygen moving into the cell. Okay? Any questions about that? Facilitated diffusion is similar, where a molecule is moving into the cell by diffusion, but it is using a protein, not really a transport protein, because the cell is not spending energy to move the molecule across. It is just acting like a channel, creating a hole in the membrane for the protein, the molecule to move from areas of high concentration to areas of low concentration. And you can see that they've called this glucose. I don't want to talk about glucose because glucose doesn't always work as facilitated diffusion. So I wanted to use the word fructose. That would be a much better example. And the reason why I haven't changed it is that the slide is, this picture is all one thing, so I can't really change it. Fructose moves into the cell only by facilitated diffusion. So when you eat fruit and there's a high concentration of fructose in the gut, the fructose moves into the gut cell by facilitated diffusion. There's a channel protein that allows the fructose to move into the cell. And what's important about this is that you will never digest all of the fructose, because once the concentration of fructose molecules inside the cell equals the concentration of the molecules outside the cell, there will be no net gain of molecules moving into the cell. So was that clear? So when you eat fructose, you will never digest all of the fructose, because once the fructose concentration is equal outside the cell, then inside the cell, no further fructose will move into the cell by a net gain, meaning you might have one move in if one moves out, but that's a net gain of the same. Now the cell can do things to make more fructose move into the cell, and that is the cell can put the fructose into the blood can take it to the liver can convert the fructose into glucose, and that would lower the fructose concentration inside the cell, so that will allow more fructose in the gut to move into the gut cell. Any question about any of that? Anyways, the important part is that fructose moves into cells by facilitated diffusion. The fructose needs a channel protein to move into the cell. And the cell does not spend energy to move fructose into the cell. And if you want to know about glucose, I'll just briefly state it. You don't need to know this. I'm not going to test you on it. Glucose will move into the cell by facilitated diffusion when the glucose is higher outside the cell than inside the cell. But when the glucose is now equal outside the cell and inside the cell changes, and the protein that allowed glucose to enter the cell by facilitated diffusion now becomes an active transport protein, and the cell spends energy to move a molecule of glucose against its concentration gradient inside the cell. So that when you eat glucose, all of the glucose that you eat will be digested and brought into the gut cells. Okay? And that's kind of complicated because glucose will act as facilitated diffusion when the glucose is higher in concentration outside the cell than inside the cell. But once it becomes equal, the protein changes and then it becomes an active transport protein, and it'll pump glucose into the cell against its concentration gradient, and the cell will do that by spending an energy of ATP to move a molecule of glucose into the cell. Okay? Like I said, you don't need to know that. I won't test you on that. So as I stated, with both simple and facilitated diffusion, solutes are transported down their concentration gradient from areas of high concentration to areas of low concentration. And both simple diffusion and facilitated diffusion, the cell gets the movement of these molecules without spending any energy. Okay? Now, osmosis is similar in the sense that the molecules are moving down their concentration gradient, and the molecules are moving without the expenditure of any energy by the cell. But with osmosis, we're talking about the movement of water, not a solute, across a selectively permeable membrane. And the water molecules are moving from areas of high water concentration to areas of low concentration. Now, please understand that we do not define tonicity by the concentration of the water molecule. We define tonicity by the concentration of the solute molecules in the water molecules. So if water is moving into the cell, that means the cell has a higher solute concentration inside the cell and a lower solute concentration outside the cell. And if you ever get confused on that, just remember the concentration of water is opposite to the concentration of the solute dissolved in the water. Is that clear? Anyone have any questions about that? So when we're talking about osmotic solutions, a bacterial cell can experience three kinds of osmotic solutions. The cell can be in an isotonic solution, a hypotonic solution, or a hypertonic solution. Now you should come to understand that these terms are relative terms. So it's correct to say that the solution outside the cell was hypertonic relative to the solution inside the cell. Of course, with isotonic, it doesn't matter because they're equal, but when we're talking about hypotonic, one solution is hypotonic relative to the solution inside the cell. So the easiest one is talking about isotonic solutions. isotonic solutions, the solute concentration outside the cell is equal to the solute concentration inside the cell. And even though we've defined tonicity as the solute concentration, it is the water that moves. And we say there's no net movement of water when the solutions are isotonic, meaning there's no net movement of water moving into the cell or out of the cell. By no net movement, if one water molecule moves into the cell, then there has to be one water molecule moving out of the cell, and therefore we say there's no net movement of water into the cell. And the cell is happy with the smiling face because this is the tonicity that cells want to be in, because if they're in an isotonic solution, they're not going to die. Okay? Any questions about that? The cell can also be in a hypotonic solution, and that is the solute concentration outside the cell is less than the solute concentration inside the cell. That means that water is higher outside the cell than inside the cell, so water will tend to move into the cell. If the cell does not have a cell wall, the cell will swell, and then the cell membrane will eventually burst. And when that happens, the cytoplasm will leak out of the cell, and then the cell will die. If the cell has a cell wall and the cell wall is intact, then water can only move into the cell up until the cell membrane moves up to the cell wall. Once the cell membrane has moved up to the cell wall, it can't move any further because the cell wall protects the cell, and no further water can move into the cell, even if the water is higher outside the cell than inside the cell. And we discussed this earlier, and that is the cell wall can protect specifically bacteria cells from osmotic lysis. But if the cell wall is damaged, like with a lysozyme or with penicillin G, then the cell membrane can leak out of the hole in the cell wall, and it'll protrude, sort of like when you're blowing a balloon in a box and the box has a hole in it, the balloon will start popping out the hole, and if you keep blowing on the balloon, it'll pop out of the hole of the box higher and higher until the balloon will eventually break. And that's what happens with the cell membrane when the cell wall is damaged and the cell is in a hypotonic solution. Most bacteria live in a hypotonic solution, and that's because they have lots of solutes inside their cell, like lots of sugars and lots of amino acid and lots of other molecules. And so bacteria tend to live in a hypotonic solution, and that is why the antibiotics like penicillin G are lethal to bacteria. Any questions about any of that? If not, let's go on to the third tonicity, hypertonic solution. The solute concentration outside the cells in a hypertonic solution is higher outside the cell than inside the cell. So water will move out of the cell because there's more water inside the cell than outside the cell. That will lead to shrinkage of the cytoplasm, and the cell membrane will pull away from the cell wall, as shown here. Let me blow that up a little bit. There's the cell membrane. It's pulled away from the cell wall because water is leaving the cell. This will eventually lead to plasmolysis, and then that will kill the cell. It generally leads to cell death unless the cell is an endospore. This will lead to cell death. And that's why you can have a bottle of molasses at room temperature for over a year or more. And when the bacteria fall in, they die by plasmolysis because the water leaves the cells. If you have any question about that, we figured that my mother had a bottle of molasses that must have been at least 20 years old, and I made molasses candy out of it, and my father ate it and loved it, and he was never harmed by it, okay? And that bottle of molasses was at least 20 years old before I made the candy out of it. And that bottle of molasses stayed around for 30 years before it was finally thrown away. And it looked perfectly well, although I never made anything out of it after 20 years old, okay? So why a hypertonic solution can keep bacteria from growing in it and then causing the bottle of molasses to spoil. Any questions about that? All right, that's the 3 tonicities. And that's all good for cells. But we do need active transport, such as when a movement of a molecule occurs against its concentration gradient. And that has to happen because passive processes are okay when the concentration of substance required by a cell is higher outside the cell than inside the cell. But what if the cell is in an environment with a low nutrient concentration, such as a bacteria cell in the open ocean? There aren't very many nutrients in ocean water. Or when we're talking about a cell in the desert, I don't know where that desert is. Let me see if I can blow that up and read it. It looks very deserty. We can't see it. Can I? Go back. That's in the way. Maybe I can get it away. Nope, it doesn't say where it's from. But anyways, the concentration of nutrients in this environment will be very low and will be much lower outside the cell than inside the cell because the cell is going to want to have nutrients inside it. And so what's the cell to do? Well, that's why they use active transport. The cell must accumulate nutrients against a concentration gradient. And the cell can do that, but it requires the cell to spend energy to move a molecule against its concentration gradient. And that's because the cell is doing work, moving the molecule against its concentration gradient. The cell uses an active transport to pump the solute molecule against its concentration gradient. There are a couple of things that are required. Move that over here now so we can see that. The cell needs a transport protein in the cell membrane, which is generally molecule specific, meaning a transport protein generally moves one and only one molecule across the cell membrane. Like there's a transport protein for moving glucose across the cell membrane, a transport protein for moving galactose across the cell membrane. And that's the only molecule that it moves. Any questions about that? How the molecule moves across its concentration gradient from areas of low concentration to areas of high concentration is the cell spends a molecule of ATP to move the molecule against its concentration gradient. So each time the molecule moves, the cell spends one molecule of ATP. All right. Any question about the active transport? Now, prokaryotes, and only prokaryotes, have a special form of active transport, meaning eukaryotic cells do not have this. We call it group translocation. It's active transport, meaning it does require a transport protein. It does cost the cell one high energy phosphate compound, such as PEP. The molecule is transported from areas of high concentration to areas of low concentration. What is different is when all of that happens, the molecule that is moved into the cell, like glucose, when it's moved by group translocation, the molecule is chemically altered, where a phosphate molecule, generally from PEP, is transferred from PEP. The phosphate group is transferred to the molecule moved across. And I'm giving the example of glucose. So inside the cell, outside the cell, you had glucose, and it'll be actively transported into the cell by group translocation. And then the glucose molecule is chemically altered. There will be a phosphate group attached to it. And so we call it glucose something phosphate. Is it isophosphate? We'll just call it glucose 6-phosphate. I don't remember what it's called, but it's something like that. Why the cell probably has this is that if the cell were to continually add glucose into the cell, the glucose inside the cell would be twice as high compared to outside the cell. Then it would be with group translocation, meaning with group translocation, the cell could actively transport glucose into the cell. And I don't know, let's say there's 5 molecules of glucose inside the cell and 1 outside the cell. And then the molecule of glucose can be moved into the cell by group translocation. So instead of glucose being in the cell, it'll be glucose 6-phosphate. And so that lowers the glucose concentration inside the cell. So instead of having 10 glucose inside the cell, you only have 5 glucose inside the cell. Is that clear? Sort of. Also, when you chemically alter the glucose, you can help prevent the escape of the molecule from the cell. But mainly it allows the cell to accumulate both glucose and glucose 6-phosphate, and that way the glucose concentration can be half the concentration inside the cell than it is outside the cell. And the reason being is that if the glucose concentration were twice that, meaning 10, it costs the cell, it's much more difficult moving the molecule when it's 1 outside the cell and 10 inside the cell than it is when it's 1 outside the cell and then 5 inside the cell. And it still costs 1 ATP, but it is harder moving against 10 than it is to move against 5. And so bacteria have this special active transport, and it's called group translocation, where the molecule is chemically changed once it is transported into the cell. Any questions about group translocation? Okay. So found only in prokaryotes, and then the molecule that's transported into the cell by group translocation is chemically altered. It gets a phosphate group attached to it. All right. Since there's no questions about group translocation, let's turn to the cytoplasm. That's the substance inside the cell membrane. It's mostly water. Generally, about 80% of the cytoplasm is water. The other 20% are the dissolved solutes in the water. And normally speaking, for most of the time, inside the cytoplasm of a bacteria, you'll have the bacteria DNA, meaning the chromosome, dissolved in the cytoplasm, meaning that bacteria chromosome is not precipitated out of the cytoplasm. It's actually dissolved in it. The only reason why we can see where the bacteria DNA is the DNA can stain differently with certain stains from the rest of the cytoplasm. Also found in the dissolved cytoplasm is all the molecules dissolved in it, like glucose, sodium chloride, the ribosomes, which these are so large that they are not dissolved in the cytoplasm, meaning they, you could call them precipitated out of the cytoplasm. You can actually see them, and I've got this blown up probably as good as I can get it. Nope, I can get it bigger. We have a dot for the artist's rendition, and if you look over here, you can sort of see dots over here, and those are the actual ribosomes in the cell. So they are not dissolved in the cytoplasm. They are so large that they actually do not dissolve in the cytoplasm. That's how I'll word it. There's the nucleoid region, and the DNA is dissolved in the cytoplasm, but it stains differently, and wherever the DNA floats around in the cytoplasm, that is where the nucleoid region is. Okay. Also, as far as DNA goes, you can have the plasmid dissolved in the cytoplasm. And then inclusion bodies, which we'll talk about more. The cell is bringing in substances inside the cell. And initially, when the substances are low in concentration, they will be dissolved inside the cytoplasm. But as the cell brings in more and more of that one substance, enough of them get together that they precipitate out of the cytoplasm. And that results in the formation of an inclusion body, where we see a precipitate in the cytoplasm, and that is called an inclusion or an inclusion body. Any questions about that?
What's the really dark spot on the microscope one?
You're talking about this one right here? It's not labeled, so I'm not sure. That might be another inclusion body. Let me see if I can blow that up. Yeah, it's not an organelle because this is a prokaryotic cell, so it doesn't have organelles. I'm guessing that it's another inclusion body, meaning this one here and that one there is an inclusion body, but so is that one. And my guess is that's what that is. This is an inclusion body, and this is an inclusion body. I'm not sure if this is another inclusion body or just a dark staining region. that might be another inclusion body.
All right, thank you.
All right. Come on. There we go. Oh, I already talked about the nucleoid region. It will be wherever the DNA floats around in the cytoplasm because there's no nuclear membrane. So you should understand that the nucleoid region has no boundary with the cytoplasm. All right, any question about that? I already talked about ribosomes, so I'm going to move on. I'll just mention that ribosomes are the site of protein synthesis in the cell, and you should know that ribosomes are made up of proteins and ribosomal RNA. So ribosomes are both protein and ribosomal RNA. And prokaryotic ribosomes are smaller than eukaryotic ribosomes. Prokaryotic ribosomes are 70S. This slide doesn't show it, but eukaryotic ribosomes are 80S. They're larger ribosomes. And I mentioned that there are inclusions in bacteria cell. They're reserve deposits within the cytoplasm, and whatever the cell is concentrating, enough of the molecule gets together that they actually precipitate out of the cytoplasm, and that is when we call it an inclusion. There are different types. You don't need to know any of the different types. I'm not going to hold you to knowing the names and what the heck they are, okay? You should just know that if you see spots in a bacteria, it's an inclusion because the bacteria have no organelles, membrane-bound organelles. I suppose one spot would be the nucleoid region, and then the others would be inclusions. Just to show you some inclusions, this cell is concentrating sulfur in it, so it metabolizes sulfur and it just pumps sulfur into the cell at high concentration that the sulfur, which isn't very soluble in water, precipitates out of the solution. Magnetosomes here and metachromatic granules here, which this is really hard to see, but in yellow, it's showing you what the cell looks like. And so each cell here has 3 metachromatic granules of inclusions. Something that I want to talk about more than inclusions are endospores. Endospores are a specialized resting cell that is produced by certain species, mainly Bacillus and Clostridium, when the environment is becoming detrimental to the survival of the parent cell. So this cell here doesn't have an endospore, but the environment is getting to be harsh. I can tell because most of these cells are producing endospores. So this cell will shortly begin starting to produce endospores because the environment is becoming detrimental. And this cell can produce endospores. Endospores are highly resistant to almost everything you can think of. They're highly resistant to desiccation, to heat, to various chemicals we use to sanitize or disinfect our objects or in our house or something like that in the lab. They're highly resistant to radiation, and they can survive conditions when the vegetative parent cell cannot. So when the environment becomes so harsh that the parent cell dies, the endospore will continue living. And that's the purpose of endospores. They are made when the cell finds a harsh environment so that the endospore can survive when the parent cell will die. And we call the parent cells vegetative cells because they grow and metabolize normally, a vegetative cell. The endospore, while it is an endospore, is a cell essentially in stasis, just waiting for the environment to become favorable. And while the cell is in an endospore, it is hardly metabolically active, meaning there's very little metabolism occurring inside an endospore. Any questions about that part? Okay. So the endospores are made when the environment becomes detrimental to the parent cell's survival. Sporulation or sporogenesis is the term used when a cell is making an endospore. Like that cell is making an endospore. This cell is making an endospore. So this is undergoing sporulation. This endospore is almost complete. It looks like it needs to be finalized right here, but other than that, it's almost complete. Germination is where the endospore, like this endospore right there, or that one right there, germinates and stops being an endospore and becomes a parent cell, like this. And then that parent cell has normal metabolism and can undergo normal growing conditions. So germination is the return to a vegetative state of an endospore. Any questions about the terminology? I'm not going to ask you the specifics of endospore formation, but the endospore follows sort of normal cellular division, and that is the DNA divides, makes two copies of itself. One copy goes to one side of the cell, the other copy goes to the other side of the cell, and then the cell divides into two. But when the cell is making an endospore, instead of dividing uniformly, meaning normally cells divide, I'm trying to get my mouse here, normally cells divide by, what is that called? Drawing the blank here.
Mitosis?
No, that's a eukaryotic cell. A bacteria cell normally divides.
Binary fission?
Say again, binary fission, thank you. Normally divides by binary fission, where the cytoplasm divides right down the middle. So 50% of the cell constituents go to one side, 50% go to the other side, and then each side develops into a distinct daughter cell. Well, in endospore formation, both cells get two copies of DNA, but the cell doesn't divide equally. The endospore will be smaller than the regular cell or the other side of the cell, okay? And then it goes through a special membrane. You get a septum forming. We're not going to talk about this. Two membranes. We're not going to talk about that. Peptidoglycan is put down inside the membranes. The spore coat forms, and that's important. The spore coat is what we're seeing here. It's highly protective of the endospore. And then the parent cell dies, and then that releases the endospore. The important part is that when you make an endospore, one cell, this cell, makes one endospore, and then the parent cell dies. That's the other side, okay? And so one cell makes one cell. This is not reproduction. Hopefully you see that. reproduction is when one cell makes two cells. Or in the case of humans, where two cells, the human egg and the human sperm, make one individual, the baby. Okay? So if one cell makes one cell, that's not reproduction. This one cell continues, but we only started with one cell. So endospores are not reproduction. It is a cellular survival technique. It is not a reproductive technique. Hopefully everybody sees that. All right, let's watch a little video on endospore formation. I can get it to go. I should have clicked that while I was still talking because it's taking a long time to load. All right, let me try and walk you through that if I can. So this cell is starting to form an endospore, and obviously the green stain is dying for the endospore, showing the endospore. Any questions about that? All right. Any questions about anything we've discussed so far? I'm not gonna talk about the eukaryotic organelles because hopefully you've already studied those in your previous class. If somebody wants me to talk about those, I can, and we'll do that after I send everybody else home. But there is one more topic I want to talk about, and you probably never heard of the endosymbiotic theory. And that's my last topic for this lesson. So if there's no questions, I'll talk about the endosymbiotic theory, and then I will let you go. But for those of you who want, I can talk about the eukaryotic organelles if anyone wants that. Like I said, hopefully you learned about those in your previous class. That's why I'm not going to talk about them. The endosymbiotic theory is a theory that I want to talk about. It's a theory to explain how eukaryotes obtain mitochondria and chloroplasts. And of course, chloroplasts are only in plant cells, they're not in animal cells. But mitochondria are in both plant cells and animal cells. And they are unique among the organelles. And so I want to talk about them. And the endosymbiotic theory explains how a eukaryotic cell got mitochondria and chloroplasts. This is covered in the custom textbook on page 275 to 276. And it's in chapter 10 if you have the regular textbook. When we look at the chloroplasts and the mitochondria, they are unusual in their being organelles because they have, I'm not seeing it here. They have, oh, there it is there. They have a double membrane and they have DNA molecules within them. They have a few other things within them that we'll talk about later, but the point is they're different from all the other organelles because they have a double membrane and they have DNA within them. The endosymbiotic theory states that there once was a proto-eukaryote cell, and I should probably open up a browser so I can write in it. Anyways, there once was a prokaryotic eukaryotic cell, meaning a precursor to the eukaryotic cell, and it never had mitochondria or chloroplasts. It also had it performed its cellular reproduction, or not cellular reproduction, it performed its ATP production by glycolysis and fermentation. So the proto-eukaryote had no mitochondria and no chloroplast, obviously, and made ATP from glycolysis, probably with fermentation. All right. Where's my slide? And that's how it happened. And the eukaryotic cell eventually engulfed another bacteria. What I really should do is draw something. So bacteria engulfed by proto-eukaryote. And for some reason, the proto-eukaryote, once it engulfed the bacteria, it never initially dissolved the bacteria. So it did not eat it. The two lived together in symbiosis, at least initially. Okay, so they were living together for some reason. Maybe the proto-eukaryote was just keeping the bacteria around for a future food supply, and so it never dissolved the bacteria initially. Well, the bacteria found glucose in the cytoplasm of the proto-eukaryote. And the bacteria performed aerobic respiration. So from one glucose, it made 38 ATP molecules. The eukaryote, or proto-eukaryote, I should say, where do I have that? Here we go, right here. So I'll put it up there. The proto-eukaryote could only make from one glucose 2 ATP. Well, the bacteria, for whatever reasons, probably because life was young back then, we're talking about a long time ago, the bacteria was in the cytoplasm of the proto-eukaryote. The bacteria released some of the ATP. Now, it probably wasn't 38 ATP, but let's say half. So what is that, 19? So let's say around 19 ATP. And why the bacteria did this is, like I said, life was just early, and mistakes were made, and the bacteria wasn't terribly worried about it because it was inside the cytoplasm. So the bacteria could pull the ATP from the cytoplasm if it needed to. But anyways, the point is that the bacteria released some of the ATP, which then went into the cytoplasm of the proto-eukaryote. Now, the proto-eukaryote said something, 19 ATP, where did this come from? I lost 1 glucose, but gained 19 ATP. Good deal. Because the proto-eukaryote could only make 2 ATP from 1 molecule of glucose. And so the proto-eukaryote decided to keep the bacteria alive inside it. And then the bacteria made ATP for the proto-eukaryote cell. So now the two are living in symbiosis. And they've been living in symbiosis for so long that now we don't find the mitochondria anywhere except in the cytoplasm of eukaryotic cells. So this is the theory of the endosymbiotic, well, the endosymbiotic theory. How mitochondria became a part of the eukaryotic cell. That long time ago, a proto-eukaryote engulfed a bacteria, which then became a part of the mitochondria of the became the cytoplasm to the eukaryotic cell, and then started living in symbiosis with the eukaryotic cell. And so nowadays, you always find the two together. The eukaryotic cell has a bacteria inside of it, called the mitochondria. And the two live in symbiosis. And you don't find the mitochondria anymore outside of the cytoplasm of a eukaryotic cell. Any question about that? Now, the same thing happened with the chloroplast in a proto-plant cell. First, it gained a mitochondria for making ATP, and then it took in a chloroplast, which was a photosynthetic bacteria. And in the plant cell, the first bacteria became the mitochondria, and then the second bacteria became the chloroplast of the plant cell. So the plant cell, by this theory, gained 2 bacteria the animal cells, only one, the mitochondria. Any questions about that? Now, there's strong evidence for this theory. First, when we look at some protist cells, we notice that they live in symbiosis with a bacteria cell that they have engulfed. This one is a, what is that called? Paramecium, which is engulfed a bacteria. And now the bacteria or the algae can engage in photosynthesis for the paramecium. And there are some amoeba which only can perform fermentation, and they don't have a mitochondria, but what they do is they engulf a bacteria which can perform aerobic respiration, and then the two live in symbiosis. The bacteria makes ATP for the amoeba, and the amoeba provides the bacteria with glucose. So there's just one family of amoeba that don't have a mitochondria. And apparently after the amoeba divides, it goes out and finds bacteria which it engulfs. And the bacteria which live with the amoeba in symbiosis act like the mitochondria in other eukaryotic cells. All right. Another evidence of support is that, as I stated, the engulfed organelles have two membranes. The mitochondria has the inner mitochondrial membrane and the outer mitochondrial membrane. In the chloroplast, the inner membrane is the granum, and the outer membrane is the outer membrane of the chloroplast, meaning they have two membranes. And how can I show that? Let's go back here. If you become a phagosome inside of a cell, you will have two membranes. You'll have your cell membrane on the mitochondria, and then you will have the phagosome membrane when this organelle or this cell consumes the other organelle. So if you were taken up by the cell, you expect this cell to have two membranes, its own cellular membrane, and then a membrane of the phagosome of this cell. And as I stated, with the mitochondria and the chloroplast, they both have two membranes. And then there are some other evidence for it. Let's talk about the bottom, because the top is the strongest evidence. When you look at the prokaryotic cells, they divide by binary fission. Eukaryotic cells divide by mitosis. The mitochondria and the chloroplast, they divide by binary fission. And their binary fission is independent of the cellular division of the eukaryotic cell. When we look at the ribosomes of a prokaryotic cell, as you know, they're 70S. The ribosomes of a eukaryotic cell are 80S. The ribosomes of the mitochondria and the chloroplast are 70S. If we look at the first amino acid and protein synthesis of eukaryotic cells, AUG codes for the amino acid methionine. Well, AUG in prokaryotic cells does not code for the amino acid methionine. It codes for the amino acid formylmethionine. And in the mitochondria and the chloroplast, AUG also codes for the amino acid formylmethionine. All right. If we look at histones present on the DNA of a eukaryotic cell, then you could also say, if you look at introns, that eukaryotic cell has histones and they have introns on the DNA. And if we look at, this should really be bacteria, the prokaryotic cell lacks histones, and bacteria lack introns. The archaea have histones and introns, but we're not talking about the archaea, we're talking about the bacteria. And if we look at the mitochondria, they do not have histones and they do not have introns. And then the strongest evidence, if you look at the DNA, eukaryotic cells have linear DNA. Prokaryotic cells have one circular molecule, and mitochondria and chloroplasts have one circular molecule of DNA. And then the strongest evidence of all, if one sequences the DNA of mitochondria and chloroplast, their sequence falls in the domain bacteria. It does not fall in the domain of the DNA for eukaryotic cells. Okay? Any question about the theory of endosymbiosis? Everyone got that? All right, well, that's it for lesson 4. I'm not going to go on lesson 5 today. If anyone wants me to talk about the organelles of eukaryotic cells, I'll do that after a break. No, maybe I should just, let's not save it. All right, any questions? No questions. All right. So for those of you who are leaving, go ahead and leave. Those of you who want to come back after a break, let's take let's make it a 12-minute break. I'll be back at 2:40, and I'll talk about the organelles of eukaryotic cells. All right?
Do we have
Okay, somebody's trying to talk. You came over just jumbled. Can you try and repeat that? You want to send me a message?
Do we have lab today? The second session?
Oh,
Can
today
you
we're having
hear
lab?
me?
No,
Will
we're
do.
supposed to have lab on Monday and Wednesday. If you look at the schedule. Oh, no, you're right. You're right. We're supposed to have lab today. Thank you. Yeah, we are supposed to have lab. No, wait a minute. We're supposed to have lab on Monday and Wednesday. So we don't have lab today. Correct? So we're not having lab today because today's Tuesday. Okay, so I'll be back at 2:40 and I'll talk about the eukaryotic cells for anyone who wants it. If nobody wants it, I'll just sign off. Okay?
Are we being tested on eukaryotic cells?
Yes, it is in the rating. And it generally, it's just stuff you've had before. But yes, I'm just going to say yes.
Okay, thank you.
All right. How do I end this?